In this module, we focus on the third and final method of analyzing associations that involves looking at the difference in means across groups. To recap, when analyzing associations between variables, the type of analysis depends on the types of variables you have in the analysis. In this module, we focus on the case where one variable is non-metric and the other is metric. In SPSS, the non-metric variable is called the grouping variable and the metric variable is called the test variable. For example, if you wanted to analyze a relationship between gender and attitude towards a brand, the first is a non-metric variable with two groups, male and female, and the second is a metric variable because it is measured on an interval scale. So we could look at the difference in the mean attitude for both groups and test to see if it is statistically different. If it is, we would conclude that there was an association between the variables gender and attitude. Similarly, we could use this approach when we want to test whether expenditure on a category is related to awareness of a product. In this case, we would see whether people who are aware of the product are different in their expenditure on the category than people who are unaware. The basic idea is that we look at the mean of the metric variable for the two groups that we are comparing. The test is based on the assumption that there are two independent samples drawn from two different populations, and we test if the means from the two populations are different. The null hypothesis is that the means of the populations are equal, that is, there is no difference in the population means. This is equivalent to saying that there is no association between the variables we are analyzing, which is gender and attitude. In order to do this test, however, you have to make some assumption about the variances of the two populations. You don't need to estimate the variance, but you have to assume either that they are equal or that they are unequal. This assumption is required because part of the formula for the test depends on what you assume. The test statistic is based on the difference between the sample means x1 bar and x2 bar. The denominator in this formula depends on whether you assume equal or unequal variances. Here I've shown the formula only for unequal variances, and this depends on the standard deviations of the two groups and the sample size n1 and n2 in each group. The test statistic is distributed as a z-statistic if the sample size in both groups is greater than 30. If the sample size in either group is less than 30, it is distributed as a t-statistic. Remember that the distribution does not affect the calculation of the test statistic, only the critical value that it is compared with. As we just discussed, in order to conduct this test, you have to make some assumption about the population variance in the two groups as to whether they are equal or unequal. But in practice, how do you choose between those assumptions? Again, SPSS comes to the rescue. It does two things to help you. First, it conducts the test under both assumptions and gives you the results so you can pick whichever one you like. Second, it conducts an additional test for equality of the two variances for the populations so you can decide what the correct assumption is. The null hypothesis for that test is that the variances are equal. This test of equal variances, called Levene's test, is an F test, that is, the test statistic is distributed as an F statistic. Now, we are not going to go over this test in the class, but you don't need to know how to do the test, only how to interpret the results that SPSS shows you. And the interpretation is the same as for any other test. You look at the p-value and you compare it with alpha. If it is less than alpha, you reject the null hypothesis. In this case, the null hypothesis is that the variances are equal. So if you reject the hypothesis, you have to assume that the variances are not equal. To begin, let's examine the different parts of the SPSS output since it's a little complicated. The first two columns labeled Levene's test relate to the test of equal variances. The first column labeled F shows the value of the test statistic for that test, and you can ignore that. The next column labeled SIG shows the p-value for that test. The remaining columns relate to the test of equality of means, that is the independent samples t-test, which is what we are mainly interested in. The next three columns show the value of the t-statistic, the degrees of freedom, and the significance, two-tailed, which is the p-value for the test. Again, you can ignore everything except the p-value column. The last thing to notice is that for every variable there are two rows of results for the test of equality of means. Look at the leftmost column to see what these rows are. The top row for each variable shows the results if you assume equal variances, and the second row shows the results if you assume unequal variances. You basically have to pick one or the other depending on which assumption you think is correct. Now that we know what we are looking at, here's a sequence of steps you need to follow. Start by looking at the significance two-tailed column, which shows the p-value for the test of equal means. If for any of the variables the p-values are less than your alpha, you have a statistically significant difference in means for that variable. If not, you don't have a significant difference. In most cases, you'll find that the significance or lack thereof will be the same regardless of what you assume about the variances. That is, both p-values will typically be either less than alpha or more than alpha. This means that the assumption about variances doesn't matter for that variable. You reach the same conclusions regardless. For example, in this output, you can see that all the p-values are greater than 0.10, so there are no significant difference in means for these data. Once in a rare while, you'll find that the p-value corresponding to equal variances assumed gives you a different conclusion than the p-value for unequal variances assumed. In those cases, you'll need to refer to Levene's test to see which is the correct assumption to make. Again, if the p-value for Levene's test is less than your alpha, you would reject the hypothesis of equal variances and assume unequal variances. In that case, you would look at the results in the row labeled unequal variances. Otherwise, if the p-value for Levene's test is greater than alpha, you would look at the row labeled equal variances. This is a lot to digest, so you may want to rewind and review this slide again. If it's any consolation, it does get easier with practice. The last step in the difference in means analysis is to actually look at the means that have been shown to be statistically significant in the previous step. This basically means you would scroll up in the SPSS output to see the table with the means, as shown here. For each test or metric variable, SPSS would show you the mean value for each group, and if the statistical test showed the means to be significantly different, you would simply note which mean was higher, and you would interpret that. For example, let's say, hypothetically, that the last variable shown here, National maintains error-free records, was found to be statistically significant using the process we discussed in the last slide. In that case, you would note that for males, the mean was higher, or 5.50, than for females, which is 5.42. In other words, males were more likely to perceive National as error-free. And that's it for this section on analysis of associations. Happy analyzing, and may all your hypotheses be supported.